A-Level Biology 9700 Paper 4 Variant 1 May June 2025. Today we are going to discuss the question number 7, and question number 7 is: insulin is an example of cell signaling molecules of the endocrine system. Outline why insulin can be described as an example of cell signaling molecule of the endocrine system. So endocrine system consists of the ductless glands. So ductless glands. Ductless glands don't have ducts, due to which they directly pour their secretions into the blood, and cell signaling is the way of communication between the cells. So, outlined by insulin can be described as an example of cell signaling molecule of the endocrine system. The first, we can say that it acts as an example because the insulin is produced and secreted directly into the bloodstream by the beta cells in the islets of Langerhans of the pancreas, which functions as an endocrine gland. So it also travels through the blood throughout the to reach its target cells, and it also acts that on the cells that are directly from its sites of protection, primarily liver, muscle, and fats cells as well. So it also binds to a specific receptor proteins on the surface of these target cells. So this binding initiates a signal transduction pathway that triggers a specific response, which is the uptake and storage of glucose, effectively it lowering the blood glucose level. So in this way, I would like to say that it is an example of cell signaling molecule. So this explanation can be written in this way. The first point which proves is that it is uh, an example of cell signaling. It is produced and secreted by islets of Langerhans of the pancreas, which acts as a ductless gland. Via blood reach to target cells. Third, acts on cells that are distant from its site of production. It binds to specific receptor proteins on the surface target cells, on the surface of target cells, which initiates a signal transduction pathway. That triggers a specific response. So, in this way, I would like to say insulin acts as a hormone, a chemical messenger which is secreted by the endocrine gland into the blood to regulate activity in distant target cells, which is the definition of endocrine signaling. And next question is, researchers compared the effect of two types of meal on the mean blood glucose concentration and the mean blood insulin concentration of 12 volunteers. The measurements were made by 3 hours after the meal. Meal A contains 55% carbohydrates, 27% fat, and 18% protein. So meal B contains 3% carbohydrates, 90% fat, and 7% protein. So figure 7.1 shows the results. And you can see the results in both these two graphs. Now, the question is, with reference to figure 7.1, explain how negative feedback operates to control means blood glucose concentration in the volunteers who ate meal A. And here you can see both types of meals. First of all, what is negative feedback, which we have to explain. So negative feedback is a mechanism that acts to reverse a change in an internal condition, bringing the condition back towards the set point. Set point means the normal range. So here in this case, you can see that they used 12 volunteers and they make use of two types of meals. Meal A contains 55% carbohydrates, means the lot of carbohydrates, and the meal B contains 3% carbohydrates. The meal A has been represented with a light color, gray color, and meal B has been represented with the dark color. So in the first graph, you can see that the mean blood glucose concentration per 100 milligram per 100 cubic centimeter. So you can see, first of all, at zero, the glucose concentration was about this point. So this point shows the normal or the set point of the glucose concentration in the body. But after eating this meal, it has been spiked that it has reached to maximum 110, and then it comes back to the normal condition in your less than normal condition. So I would like to explain this. After consuming meal A, the food is digested and the glucose is absorbed into the bloodstream, which shows the mean blood glucose concentration. Already told you that is 88. It reached to 100 and then 100 and then more than 110 milligram per 100 cubic centimeters. So this increases the deviation from the normal set point. This normal set point was about 88 in this case. So the rise in blood glucose is detected by the beta cells in the islet of Langerhans in the pancreas. So the pancreas secretes the hormone insulin into the bloodstream. So where the mean blood insulin concentration rises sharply from approximately 300. So it was 300 and then and in the second graph you can see the mean blood insulin concentration. So in normal condition at zero, it was approximately 300. And then it reached up to the point which is 500. So it means when that those volunteers ate the food consist of meal A, which were mostly the carbohydrates. That is the more than 50% carbohydrates. So it spiked, it reached to the maximum level. Then insulin act on target cells, liver, muscles, and stimulating them. So in this way, then glucose from convert excess glucose into the storage polysaccharide, increase glucose use in cellular respiration. So these actions of insulin, that is respiration and the storage, glucose is converted into glycogen. So the process of glycogenesis takes place over here. And due to this increase glucose use in cellular respiration, this action of insulin caused the blood glucose concentration to fall. That is, it was fall from 500 to reach to the less than 300 again. And over here on this case, it is reached below than 80. So around 60 minutes. So it took 60 minutes to become normal. So this decrease in blood glucose concentration negatively feeds back to the pancreas, causing the rate of insulin secretion to fall, which is seen as the insulin concentration drops after 30 seconds. So here you can see it is stopped after 30 times. So I would like to say this process ensures that the elevated blood glucose level is reversed and brought back down to the normal homeostatic range. The process of blood sugar regulation is explained in this question. So after this explanation, I would like to write the answer on the given space. I have explained it on the diagram with the help of this figure 7.1. So I'm going to write the answer on the given space. First of all, I'm going over here, that is negative feedback has been shown over here. So we will go first of all, after consuming meal A, the food is digested and absorbed. And absorbed shown in figure 7.1. the increased level of glucose from 88 to 110 milligram percent. So this increase is the deviation from normal set point. From normal set point. The rise is detected by beta cells islets of Langerhans from pancreas which will secretes insulin insulin as its concentration is increasing from 300 milligram to 500 milligram on graph. It will convert the excess glucose into glycogen. Due to which concentration of glucose fall back to set point. After 30 minutes, shown into the graph. It is an example of negative feedback. Okay, now the last part of this question is suggest explanations for the changes in mean blood glucose concentration of the volunteers who ate meal B. So we will move back towards the graph and on you can see who ate the meal B. So meal B, you can see it was consist of very small percentage of carbohydrates, only 3% carbohydrates. 90% fat and 7% protein. So here we are going to again observe the, you can see glucose level at zero, it is about set point. And after eating the meal, it has been dropped after 30 minutes, 60 minutes, it is dropped to less than the normal point. And on the other hand, the glucose insulin which was secreted during this was very small quantity of the insulin was secreted during this. So here what happened, that is their fat slows absorption, was very high fat contents in this, due to which slows down gastric emptying. So small amount of carbohydrates present is absorbed much more slowly, preventing a quick spike. So the blood glucose level remains relatively low and stable, fluctuating narrowingly between 80 and 85 after the initial drop. So low insulin response due to the minimal rise in blood glucose, the pancreas is not significantly stimulated and the insulin concentration remains low and steady. As the blood glucose concentration drops, the alpha cells of pancreas are stimulated to secrete the hormone glucagon. So glucagon triggers the liver to maintain blood glucose through two processes. This is glyco, glycogen lysis, breakdown of stored glycogen into glucose. So action of glucagon counteracts this slight drop, ensuring that the blood glucose concentration is maintained within the healthy homeostatic range via negative feedback despite the lack of dietary carbohydrates. So here you can see we will move to the place where we have to write the answer. So we will start the answer in this way. Here, first of all, the graph shows a slight initial drop in mean blood glucose from 88 to 80 milligram per 100. So it means because meat contains negligible amount of carbohydrates. So the very high content fats are very high content of meat B, which will also slows down slows down the absorption of carbohydrates. The blood glucose level remains relatively low and stable. As its concentration is dropped, the alpha cells of the alpha cells of pancreas are stimulated to secrete glucagon, which triggers the liver, triggers the liver to maintain blood glucose to glucoglycolysis. That is the breakdown of glycogen into glucose. Which ensures the well, it was all about question number 7. Please like, share, and subscribe my channel to watch more videos on the papers. Thanks for watching.